Continuing our discussion here of the sensory system, how intensity and duration is encoded. So again, intensity is encoded by amount of action potentials, how many are being fired, and duration is, well, how long these action potentials are being fired. There are two different types of the duration encoding, and that will be tonic receptors versus phasic receptors. Tonic receptors, they keep firing no matter what. As long as the stimulus is there, they keep firing action potentials, and you know it. Pain receptors are a good example for that. Now, phasic receptors, they stop firing even though the stimulus is still there. Good example for that would be your olfactory system. Think about the famous garlic room you're entering in. At first it smells real strong, and then it seems like as if it doesn't smell at all anymore, because these are phasic receptors, and they will fire one more time at the end when a stimulus is withdrawn, for example. So here will be a diagram showing this, these different modalities. You can tell that in this row right here, you have fewer action potentials being fired, so that's a smaller stimulus. Here is a longer and stronger stimulus, and that means you're going to have a lot more action potentials that are being fired. Okay, so tonic receptors here, one more time. Tonic receptors, they keep firing. They have a constant firing rate, so as long as the stimulus is there, they just keep firing. And phasic receptors, think about phasing out. The stimulus is still there, right there, but there's no action potentials being fired. So down here, no action potentials fired. The stimulus is still there, but they phased out. They don't fire anymore. And to you, the perception is as if the stimulus is no longer there. A lot of receptors, especially like olfactory receptors, they are all phasic receptors. Okay, so here. Now, sort of the different modalities of the somatic senses that we're going to briefly go through will be touch, proprioception, temperature, nociception, that would be pain and itch type of perception. Now, if you did the lab last week, you should have tried some of these out, and everybody can relate to touch and temperature, pain receptors. We have all experienced that. So here is the pathway for somatic senses. You have your receptors down here, and of course these are all afferent, afferent pathways. We're having the stimulus is somewhere perceived by some sort of a sensory receptor. Then we're going toward the central nervous system, entering the spinal cord, and then using ascending tracks, and then we're ending up somewhere here on the somatosensory cortex, the slice here of cortex that evaluates those somatic senses. Now, this slice of somatosensory cortex in their brain, and that one, it's kind of funny. It basically reserves space by density of receptors. So areas of your body that have high density of receptors, like your fingertip, it seems to be overrepresented because it's a fairly small area, but it gets a pretty large slice here or spot on the cortex. Your lips are also kind of overrepresented because they're highly sensitive, have lots of receptors, and so you get an a larger area of cortex reserved for that. Now, your thigh area is a big part of your body, but it gets only a relatively small area of cortex. So it's all by density of receptors, and this thing is referred to as a homunculus, like a distorted human being, basically, where the human body is sort of represented by the density of the receptors that are there. And again, these are the somatosensory receptors. So here, another figure, a table that kind of summarizes the types of receptors, the stimulus that they process, the location, the structure, and the adaptation. You can read through that. Let's take a look at your skin real quick. You have various types of receptors in here. There are touch receptors, then there are pressure receptors, Meissner's corpuscles. Here will be a little pressure receptor. And so you can see the different types of information, pain, touch, pressure, hot and cold, all of this will be process to these nerve ends that are here and transporting information then toward the somatosensory cortex. So here a little bit more information about some of these somatic senses that we have. Starting here with the temperature receptors. They're free nerve endings. They are underneath your skin, and you do have cold and warm or hot receptors. The cold receptors measure any kind of temperatures that are lower than body temperature, and the warm receptors, they will respond to temperatures above about 45 degrees Celsius. So that's really very high fever. So remember, body temperature is about 37 degrees Celsius. So 45, it gets warm. And once the temperature is so hot that it starts hurting, then you're activating the pain receptors to perceive the heat. And that's supposed to be protecting you from dangerous kind of heat that could denature your proteins. Nociceptors, those are pain receptors. They respond to strong, noxious stimuli that could be damaging to your body. They're also free nerve ends, and they're really an alarm system. They're alerting you to painful kind of stuff that could be damaging. And they could be modulated by some chemical, substance P, that's a strong painkiller that could be could be sent out to sort of stop the perception of strong pain. The nociceptors also mediate inflammatory responses. That's why when you have an inflammation going on somewhere, that's you perceive that a lot of times as pain, so inflammatory pain. A little bit more here on these nociceptors. They are really supposed to be protecting you. They're alerting you to a problem, and they're supposed to protect you from, potentially damaging kind of events. They are integrated with the spinal cord, and they a lot of times give you this withdrawal reflex. Think about it. If you were to step on a push pin, for example, and that hurts, so then you have this withdrawal reflex. Or when you're touching a hot plate, then you have this withdrawal reflex. Very fast movement of your muscles to get you away from this painful stimulus. Of course, they're using ascending pathways to the cerebral cortex, and the sensation is either perceived as pain or as itch. So I skipped over a few slides. You can take a look at those, but we don't really need the specific type of fibers for this course. But I do want to say a little bit about painkillers. Those are analgesic drugs, and there's this whole class of non-steroidal anti-inflammatory drugs, those NSAIDs. The first one being aspirin. Anytime we have analgesic, when you hear that term, that's pain killing. And the way these non-steroidal anti-inflammatory drugs work is by inhibiting prostaglandins. And prostaglandins, that is the substance that's released from tissues that are either injured or inflamed. And so these prostaglandins, they are local tissue hormones that mediate inflammation and also pain, sort of to get your attention to the area that has a problem. And of course, if you are taking something that inhibits prostaglandins, well, then you also reduce the inflammation, you reduce the pain along with it. Opioids are much stronger substances, and they block pain perception. So they're decreasing primary sensory neurotransmitter release, and thereby blocking basically the transmission of pain. Now, there we have natural opioids, so-called endorphins. You may have heard of something like the runner's high. Running is kind of painful. And after about 10 minutes, all of a sudden it gets really easy. And that's because endogenous opioids, as endorphins, are being released, and you don't really feel the pain anymore. They block the pain. Now, you also know that opioids and the derivatives, they're highly addictive because people feel so good on them. But then if you're taking a lot of them, then they downregulate the receptors, and then you need more and more of it, and there can be a big problem because they're also central nervous system depressants, and they can be, well, it can be deadly if somebody does too much of these opioids. Now, something about referred pain here. Sometimes when you actually have pain in an internal organ, for example, let's take this here, liver and gallbladder. So let's say this area is actually hurting and sending signals that something is painful and not right. You may perceive that as something that's hurting here on your shoulder. There's different other examples here too. The reason for that is, let's take a look at the next slide here. The reason for that is that sometimes these nerves, they run in parallel, they run together. So here will be the kidney. Let's say the kidney is hurting and is sending signals along this pathway, and you can see how the skin, this patch of skin, the nerve that is reporting from this patch of skin, is running very close together. And so this is the usual stimulus. The skin here, that hurts much more often here than the kidneys. And so since they're running very close together, your brain may make a mistake and think, oh, it's got to come from the skin, because that's where it's usually coming from and not the kidneys. And so that's referred to as referred pain. You may have also heard of something similar, and that's phantom pain, where somebody with an amputated leg swears that their big toe is itching when they don't even have that big toe anymore. So that's for a similar reason, because sometimes these nerves, they run very close together. And then your brain ends up misinterpreting where these actual potentials actually came from. So that concludes our discussion of the general senses. Now we're moving on to special senses. The first one that we have here is the olfactory system. Believe it or not, your olfactory system is actually really good. The only problem with our olfactory system is that it's kind of in a bad location. So our olfactory epithelium is sitting deep in the roof of your nose. So you have this right here, this little box, and it's enlarged, and you can see this is the roof of your nose right there. This is where your olfactory epithelium is. So any odorant molecules that you want to perceive, they would have to first enter your nose and just hit all the way right here. Now, it's not too hard to do, but you all know that if you have a bad cold and you have a stuffy nose and everything is swollen up here, and the odorant molecules would no longer be able to reach this olfactory epithelium, you know what happens. Nothing tastes the same. You feel like there's just no perception, and that's basically what it is. Keep in mind that most of what we think we taste, we actually smell. And that's why when you have a bad cold, nothing tastes the same. Well, it still tastes the same, but it doesn't smell the same. And we a lot of time confuse olfactory perception with your taste perception. But at any rate, let's take a look at this here. So your olfactory system is sitting right here. The olfactory epithelium is very sensitive. These are thousands of G protein coupled receptors that then they transmit their signals, these actual potentials, to cranial nerve number 1, which is the olfactory bulb. Then we go olfactory tract, and then olfactory cortex for evaluation area. You can see the proximity of all of this. It's very close together, therefore very fast, and our olfactory perception is actually excellent. You also have this very convenient pathway right here through the ethmoid bone. You might remember from anatomy, these are the olfactory foramina that accommodate these nerves to go through and synapse right here to the olfactory bulb. So the pathway is olfactory neurons here, and there's olfactory epithelium, cranial nerve number 1 right here, then the olfactory bulb, olfactory tract, olfactory cortex, and then you have your perception right there. Also, one word about this, the limbic system here is involved. Limbic system, that's emotions. And so a lot of times what happens is that you end up having emotional events linked to certain smells. many people, when they smell something after a long time, it will bring memories back from the place and the events surrounding the first time that they smelled something. So it's basically linked to a memory as well, your olfactory system.